What was the era before dinosaurs truly like? Paleozoic, Mesozoic, etc. Beneath a sky unrecognizable to our eyes, vast oceans churned with strange armored fish, while towering forests of primitive plants clung to fragile coastlines. This was the world long before dinosaurs claimed dominion. A planet in flux where mass extinctions and climatic upheavals shaped life's possibilities. Mountains rose and fell, rivers carved new paths, and the first creatures began to crawl toward land, testing the boundaries of survival. In the shadows of these ancient landscapes, evolution quietly rehearsed its greatest dramas. Step with us into this forgotten past and witness the earth before its giants roamed. Before the roar, the world dinosaurs never saw, the earliest chapters of Earth's deep history unfolded across landscapes that bore no resemblance to the world that dinosaurs would one day dominate. As continents slowly assembled from molten fragments and oceans gathered chemical gradients that shaped the first biological experiments. Geological studies using zircon dating show that continental crust continued to stabilize between 700 and 550 million years ago, forming coastlines that shifted with each pulse of tectonic heat. These fractured regions nurtured shallow seas where oxygen remained scarce, creating environments in which early multicellular organisms navigated chemical conditions far more unstable than anything seen in later eras. Microbial mats blanketed the seafloor in wide, wrinkled expanses, recording fluctuations in metals like molybdenum and uranium that reveal alternating oxic and anoxic intervals, each imposing new evolutionary pressures. Within these fluctuating waters, the earliest macroscopic organisms developed body plans that challenge modern classification, displaying shapes that seem more mathematical than anatomical. Dickinsonia spread across microbial mats in broad, ribbed ovals, absorbing nutrients directly through its surface in a feeding strategy suited to oxygen-poor seas. Charnia and Rengeia rose from the substrate like branching fronds, their fractal patterns shaped by the flow of currents rather than rigid skeletons or articulated limbs. Fossils from the Ediacara Hills and White Sea preserve these forms in exquisite detail, showing impressions so delicate that microbial textures remain visible on the rock surfaces. Geochemical signatures suggest that atmospheric oxygen may have hovered near only 10% of modern levels, reinforcing the idea that soft-bodied, diffusion-based organisms were the only viable inhabitants at this time. As continents shifted and new shallow shelves formed, evolutionary experimentation gained momentum, allowing the earliest hints of movement, predation, and coordinated behavior to appear long before the emergence of jaws or limbs. Trace fossils attributed to Yorgia and Kimberella show looping paths that mark deliberate grazing behaviors, indicating that these organisms possessed muscular undersides capable of slow, directional travel. Kimberella's feeding structure, sometimes compared to a primitive radula, left characteristic scrape marks that suggest controlled foraging across thin microbial substrates. Meanwhile, narrow burrow-like traces assigned to bilaterian animals reveal the arrival of organisms with front-back orientation, primitive nervous systems, and the ability to disturb sediment layers in purposeful ways. These behaviors introduced subtle ecological changes, initiating feedback loops that would later accelerate the diversification seen in the Cambrian. Farther from shore, the deep oceans maintained conditions that limited the spread of complex life, yet they played an essential role in preparing Earth for biological expansion. Iron speciation analyses from ancient black shales show that deeper waters remained largely anoxic, confining metabolically demanding organisms to the well-lit shallows. Tectonic rifting associated with the breakup of Rodinia opened new ocean passages, altering nutrient circulation and influencing the distribution of essential elements like phosphorus and iron. Glacial events, such as the Gasquier's glaciation around 580 million years ago, punctuated these changes, dropping stones into marine sediments and generating cooling intervals that disrupted ecosystems in ways detectable in global stratigraphic records. These environmental oscillations may have acted as evolutionary catalysts, forcing organisms to adapt to rapid climate swings and shifting chemical gradients. Along emerging coastlines, microbial communities shaped ecological structure with remarkable influence, creating stable substrates that supported early macroscopic life while driving biochemical cycles that altered the atmosphere. Stromatolite reefs, built by cyanobacteria, trapped sediments and produced oxygen, contributing to the slow rise of atmospheric O2 that paved the way for larger, more energetic organisms. Wrinkle mats and demole structures reveal how microbial films stabilize sediments, enabling sessile organisms to anchor themselves and create vertical, ecological layering long before animals developed true reef-building capabilities. Carbon and sulfur isotope data from these deposits capture the metabolic signatures of large-scale organic burial events, indicating that microbial productivity played a crucial role in regulating climate and oxygen levels during this period. These changes gradually created environmental thresholds that permitted more complex metabolisms to evolve. In shallow marine habitats, ecological interactions followed rules fundamentally different from those that would later govern the age of vertebrates and giant reptiles. Organisms competed for space rather than pursuing or escaping predators, optimizing their shapes to maximize exposure to water currents and dissolved nutrients. Frond-like organisms adopted branching or radial forms that increased surface area and enhanced passive feeding in stable, undisturbed sediments. Because bioturbation remained minimal, the sea floor retained laminated layers that preserved fossils with exceptional clarity, while also maintaining ecological stability that limited disruptive change. Only when early bilaterian animals began burrowing more actively did oxygen penetrate deeper into the sediment, creating new ecological zones that would eventually support more aggressive and mobile forms of life. Across the ancient ocean floor, these increases in oxygen and sediment disturbance initiated subtle but transformative evolutionary developments that hinted at the dramatic changes to come. Carbonate platforms document early biomineralization, including Claudina, whose tube-like calcium carbonate exoskeleton represents one of the earliest examples of protective hard tissue. The discovery of drill holes in Claudina fossils suggests that predation had begun, indicating that organisms were starting to evolve strategies for attack and defense. These innovations accumulated slowly over millions of years, but they created conditions in which ecological roles and biological diversity could expand rapidly once environmental barriers loosened. Global stratigraphic sequences capture these subtle beginnings, showing how life prepared for an evolutionary leap even as its outward forms remained simple. Through this long and quiet era before dinosaurs existed even as concepts, Earth's oceans and continents cultivated patterns of chemistry, climate, and biology that nudged life toward increasing complexity. Each shift in oxygen levels, each pulse of tectonic activity, and each new biological experiment brought ecosystems closer to a threshold where innovation could accelerate dramatically. The gentle fronds, rippling mats, and early burrowers of this ancient world hinted at possibilities that would soon erupt with extraordinary force, reshaping environments in ways that would echo through every later chapter of Earth's story and guiding evolution toward forms far more dynamic than anything seen in these earliest seas. The Cambrian blaze, when life learned to compete. From the quiet innovations of earlier seas, a sudden proliferation of forms emerged as oxygen levels reached thresholds that fueled unprecedented biological activity. Microbial mats and simple fronds had paved the way, but now ecosystems throbbed with energy as organisms began to sense, move, and interact in ways that fundamentally altered their world. Shallow shelves and continental margins hosted creatures experimenting with propulsion, from undulating lobopodians to newly jointed appendages that allowed more deliberate navigation across the seafloor. The waters themselves became theatres of innovation, as chemical gradients, sunlight, and sediment conditions created niches ripe for exploration and exploitation. Within these newly oxygenated waters, organisms developed sensory structures that allowed them to detect movement, light, and chemical cues with remarkable precision. Trilobites evolved compound eyes, capable of perceiving motion across broad fields, granting them awareness previously unimaginable for soft-bodied predecessors. Even simple chordates, like Picaia, displayed early neural coordination, enabling dorsoventral flexion for more controlled swimming. These innovations set the stage for predation and defense, introducing pressures that drove morphological and behavioral adaptations at a pace unseen in prior eras. Each sensory breakthrough amplified evolutionary possibilities, shaping not just individual survival but entire ecological frameworks. In these crowded habitats, mobility became a defining advantage. Muscular contractions, hydrostatic skeletons, and jointed limbs allowed creatures to pursue food, escape threats, or explore new territories. Burrowers disrupted sediments, oxygenating layers previously untouched and creating new microhabitats for smaller organisms. Filter feeders extended tentacles and ciliated structures to capture nutrients from currents, while ambush predators remained concealed until sudden motion revealed their presence. Every movement left traces in the fossil record. Sinuous trails, scratch marks, and feeding depressions that tell a story of experimentation and adaptation at every scale. With mobility came confrontation, and the earliest predators introduced selective pressures that reshaped anatomy and survival strategies. Anomalocaris, one of the first apex hunters, wielded grasping frontal appendages and a circular mouth lined with serrated plates capable of slicing through soft-bodied prey. Shell-bearing organisms developed spines, armor, and mineralized exteriors to resist attacks, while small burrowers evolved rapid retraction behaviors to escape detection. Evidence of bite marks and drill holes in Cloudina fossils documents the escalating arms race, showing that predation was already a powerful force guiding morphological innovation. Each encounter between predator and prey intensified selective pressures, accelerating the diversity and complexity of life across marine ecosystems. Among the most striking innovations were early visual systems and nervous networks that allowed creatures to interpret their surroundings with unprecedented clarity. Compound eyes of trilobites could detect predators and conspecifics in near real time, while paired ganglia in lobopodians coordinated rapid, muscular responses. Neural wiring allowed precise limb control, whether for crawling along microbial substrates, gripping prey, or stabilizing body orientation in flowing currents. The evolution of these sensory and motor capabilities reshaped ecological interactions, enabling behaviors such as coordinated hunting, escape maneuvers, and territorial competition, all of which contributed to ecosystem-level complexity. New ecological roles emerged quickly as scavengers, filter feeders, ambush predators, and sediment disruptors carved out distinct niches within expanding marine landscapes. Lobopodians patrolled the sea floor, consuming microbial mats or smaller invertebrates, while radiodonts dominated mid-water hunting grounds. Trilobites diversified into numerous families, each occupying different benthic zones, from burrowers to surface grazers. Priapulids and other worm-like organisms penetrated sediments, increasing oxygenation and redistributing nutrients. The appearance of these specialized roles facilitated energy flow and trophic layering, creating a structure that allowed more species to coexist and reducing the vulnerability of ecosystems to sudden environmental changes. Hard-bodied organisms, such as trilobites, recorded their existence in durable exoskeletons that also facilitated evolutionary experimentation. Variations in segment size, spine arrangement, and body shape reflect adaptations to predation, locomotion, and resource acquisition. Cloudina and other calcified organisms offer insights into early biomineralization, preserving evidence of predatory drilling and repair, demonstrating that life was already engaged in sophisticated interactions. The fossil record preserves both individual adaptations and broader ecological patterns, revealing a marine world rapidly transforming from simple communities into dynamic, interdependent networks. Other lineages pioneered internal structural frameworks, developing supportive rods and plates that foreshadowed vertebrate skeletons. Early chordates, like Picaea, displayed notochord-based support, facilitating undulatory swimming and more complex behaviors. Segmentation in annelid-like worms enabled controlled movement through sediment, while hydrostatic skeletons in lobopodians and onychophorans provided flexible yet stable locomotion. These structural innovations increased versatility, allowing organisms to exploit new habitats, evade predators, and compete for resources in ways impossible for earlier life forms. Each incremental innovation compounded ecological opportunity, pushing ecosystems toward a new scale of complexity. On the seafloor, increasing bioturbation indicates that organisms were actively reshaping their environment. Burrowing and sediment disturbance altered nutrient distribution, oxygenation, and microbial mat structure, creating feedback loops that allowed more complex communities to persist. This environmental engineering marked a critical transition from passive occupancy to active ecosystem construction. Layers of mixed sediment, trace fossils, and feeding burrows document how life became an agent of its own habitat transformation, laying the foundations for ever more intricate interactions among species. Toward the climax of this evolutionary surge, apex predators dominated open waters, exerting selective pressures that catalyzed further diversity. Anomalocaris and other radiodonts hunted mobile prey, while defensive adaptations proliferated among benthic organisms. Armor, spines, and evasive behaviors became widespread, establishing early predator-prey arms races. The escalating competition ensured that only the most adaptable forms thrived, setting the tempo for further evolutionary radiations. Every layer of sediment, every fossilized exoskeleton, and every trace fossil records this drama in precise detail, capturing the unfolding blaze of life's ingenuity. During these escalating interactions, marine ecosystems gained stability rooted in diversity rather than uniformity. Trophic layers, habitat specialization, and the interplay of predation and defense established dynamic networks that buffered communities against environmental stress. Energy flowed efficiently through newly defined niches, and the repeated cycles of innovation reinforced the growth of complex food webs. In the wake of this evolutionary blaze, the oceans held communities far more intricate than anything the planet had known, with organisms actively shaping both their surroundings and each other. The Cambrian seas had become arenas of innovation, conflict, and cooperation, laying the foundations for the vast ecological architectures that would define the rest of the Paleozoic, kingdoms of coral and quiet titans. As the rapid experiments of the Cambrian subsided, the oceans expanded into structured arenas, alive with movement, color, and competition. Sunlight filtered through warm, shallow waters, illuminating reefs that rose like living citadels from the seafloor. Tiny grazers and filter feeders darted among the spires, each organism performing its role in an intricate dance of sustenance and survival. Currents stirred plankton blooms that shimmered like drifting clouds, carrying oxygen and nutrients to every corner of these bustling habitats. Life no longer lingered in simplicity. It moved, consumed, and interacted with a precision shaped by millions of years of evolutionary refinement. Jawless fish glided through currents with surprising agility, their cartilaginous bodies responding to chemical signals and subtle shifts in water flow. Bands of these early swimmers navigated reef passages with coordinated movements, avoiding predators and seeking out abundant microfauna. Their bodies, slender and flexible, allowed them to weave effortlessly through dense structures of bryozoans and stromatoporoids, while subtle lateral lines detected turbulence and motion from a distance. Even in the absence of jaws, these fish exhibited remarkable efficiency, brushing against the currents to sense the rhythm of the oceans and adjusting their movements accordingly. Reefs grew upward and outward, formed by colonies of early bryozoans and stromatoporoids that fused into durable frameworks. Within these layered habitats, sessile creatures like filter feeders extended feathery appendages to capture plankton drifting in the currents, while small grazers scuttled along the surfaces, scraping microbial films. Each reef spire offered shelter, feeding zones, and vantage points, creating microenvironments within microenvironments. The constant growth and structural complexity of these living architectures nurtured vertical communities, where species stacked themselves in layers that maximize light, protection, and access to food. Plankton blooms shimmered in the sunlight, their minute bodies pulsing with life and driving oxygen cycles that sustained the thriving reefs. As photosynthetic plankton multiplied, oxygen levels surged, enriching the water and enabling more active predation and movement. Nutrients carried from tidal surges and currents fueled rapid growth, connecting distant habitats through invisible threads of energy. The oceans became a network of productivity, where even small shifts in plankton density could ripple through entire communities, affecting the behavior of grazers, predators, and reef engineers alike. When microorganisms surged in massive numbers, they not only released oxygen, but also carried swarms of drifting larvae along invisible reef highways. Tiny planktonic larvae, carried by currents, colonized new reef areas and enabled rapid species dispersal across shallow seas. These microscopic travelers provided a continuous influx of juveniles for sessile organisms, replenishing colonies of bryozoans and stromatoporoids and ensuring genetic diversity. The same currents carried organic matter to deeper zones, feeding benthic detritivores and sustaining complex food webs beyond the immediate reef structures. This constant circulation of life and nutrients created a self-reinforcing system in which the health of one habitat directly influenced neighboring ecosystems, setting the stage for expansive, interconnected marine landscapes. Schools of tiny swimmers stirred currents, distributing nutrients and linking distant habitats through their collective motion. Their movements, synchronized yet unpredictable, provided both ecological stability and occasional chaos, stirring up sediment, oxygenating deeper layers, and redistributing food resources. Predators learned to track these shifting currents, honing strategies that allowed them to exploit fleeting opportunities. The oceans, once simple and static, now thrum with interaction, as every organism influences the lives of its neighbors and the chemical balance of its environment. Predatory encounters unfolded in real time, as agile hunters darted across open water, while others hid among reef spires, waiting for the perfect strike. Jawless fish and small benthic predators coordinated movement, ambushed unsuspecting grazers, or chased them across complex terrain. The interplay of stealth, speed, and sensory detection created a dynamic balance where both predator and prey were continually adapting to one another's presence. Each chase, each evasive maneuver, reshaped behavior patterns and reinforced the ecological rules of the Ordovician seas. Sensory adaptations became vital for survival. Lateral lines and primitive eyes guided fish through reefs, detecting even subtle movements among densely packed organisms. These abilities allowed creatures to navigate maze-like structures, sense approaching predators, and locate drifting plankton. Even the smallest invertebrates adjusted their feeding behavior in response to currents, light levels, or shadows cast by larger neighbors. Awareness of surroundings became as crucial as speed or strength, and ecosystems began to function as networks of perception and response, each organism attuned to both opportunity and threat. Vertical layering of the reef created stratified communities, each level hosting specialized species, exploiting light, shelter, and food in unique ways. Surface dwellers captured sunlight-driven plankton, mid-level grazers scoured microbial films, and deeper inhabitants filtered detritus from flowing currents. This spatial complexity allowed numerous species to coexist, reducing direct competition while encouraging intricate interactions. Life had moved beyond simple survival into a realm of functional diversity, where niche specialization reinforced both individual fitness and ecosystem resilience. Currents and tidal pulses carried oxygen and nutrients through every niche, linking physical processes with the living networks of the ocean. Waves stirred the reefs, circulating plankton, distributing sediment, and subtly shifting the positions of small grazers. Oxygen-rich waters supported more active lifestyles, enabling faster movement, more complex predation, and greater metabolic activity. In turn, organisms modified their environment through movement, feeding, and excretion, creating feedback loops where life itself shaped the physical and chemical structure of its habitat. Despite the apparent abundance, the seas remained fragile, and slight shifts in chemistry, temperature, or nutrient flow immediately reverberated through these intricately balanced ecosystems. Plankton productivity could fluctuate, predatory pressure could spike, and storms could disrupt reef structures, reminding all inhabitants that prosperity was contingent on constant adaptation. These subtle pressures foreshadowed the Ordovician's first major extinction events, highlighting that even thriving, diversified communities were vulnerable to rapid environmental change. The Ordovician oceans thrived as vibrant, interwoven kingdoms, their living architecture and ceaseless activity shaping both the creatures within them and the very environment that sustained life. Reefs stretched like coral fortresses, schools of fish moved in intricate patterns, and plankton blooms fueled cycles of oxygenation and energy that reached into every niche. The oceans had become dynamic tapestries of interaction, cooperation, and competition, creating stability and complexity while simultaneously hinting at the challenges that would test life's resilience in the chapters to come. Fall of the ancient seas. Even the most intricate reef kingdoms could not escape the creeping chill that began to sweep across the oceans. Sunlight cut through cooler waters at sharper angles, casting shifting patterns across reef towers and bryozoan mats. Tiny grazers and filter feeders moved hesitantly, responding to currents altered by the expansion of southern ice sheets. The pulse of the Ordovician seas slowed as temperature gradients stiffened water layers, creating pockets where oxygen sank while carbon dioxide fluctuated, subtly reshaping the chemical stage upon which life depended. Life's vibrant choreography faltered, and the bustling, interwoven networks that had defined these oceans began to fragment under the weight of a changing climate. Sea levels fell dramatically as glacial ice advanced across Gondwana, exposing continental shelves and leaving formerly submerged habitats stranded. Shallow reefs that had stretched for kilometers were reduced to isolated patches, creating disconnected communities that now had to survive independently. Currents shifted unpredictably, concentrating nutrients in some regions while starving others, and the once continuous corridors for larval dispersal narrowed into perilous channels. Species adapted for wide-ranging movement found themselves constrained, while sessile colonies faced unprecedented challenges in reproduction and growth. The ocean's physical architecture was transformed, and the once familiar patterns of ecological interaction became fragile and uncertain. Microscopic drifters that once fueled abundance now thinned, and with them, oxygen and nutrient levels began to dwindle across the seas. Phytoplankton and cyanobacterial populations collapsed in parts of the shallow waters, leaving filter feeders with fewer resources and grazing organisms scrambling for food. Oxygen pore zones expanded in low-lying basins, creating invisible barriers that restricted movement and survival. The reduction of these foundational organisms rippled through every trophic level, reshaping predator-prey dynamics and forcing communities to reorganize or face extinction. Even subtle shifts in microscopic populations now dictate the fate of entire reef ecosystems, demonstrating the delicate interdependence of life and environment. Early vertebrate pioneers and reef inhabitants found their movements restricted as currents shifted and habitats shrank under cooling seas. Small, jawless fish, adapted by altering swimming patterns, moving cautiously through reef passages to conserve energy and avoid hypoxic zones. Benthic invertebrates dug deeper into sediment layers or retreated into crevices, while filter feeders adjusted feeding rates to cope with sporadic plankton availability. Even seemingly minor behavioral shifts had cascading consequences. Predators found prey scarcer, while prey species were crowded into limited refuges, increasing competition and forcing rapid ecological recalibration. Cycles of water movement and organic material shifted drastically, creating zones too poor to sustain the once dense benthic communities. Sediment redistribution slowed in deeper areas, leaving some surfaces barren while other regions became thick with decaying organic matter. Burrowing worms, early mollusks, and small arthropods clustered in the most hospitable microhabitats, creating patchwork communities surrounded by lifeless seafloor. These refuges acted as temporary sanctuaries, allowing certain lineages to persist while simultaneously reshaping food webs and forcing species into unfamiliar interactions that accelerated adaptation. Chemical fluctuations in the oceans created pockets of unusual acidity and salinity, forcing organisms to develop tolerance strategies or abandon their habitats entirely. Minerals crucial for shell building, like calcium carbonate, became less available in some areas, slowing the growth of early corals, brachiopods, and other calcifying invertebrates. Some species shifted feeding strategies, consuming detritus or sediment particles instead of plankton, while others migrated toward localized refugia where water chemistry remained favorable. These environmental microgradients also fostered isolated pockets of genetic diversification, as small populations adapted to unique conditions, sowing the seeds for future evolutionary innovation. In the face of habitat loss, predatory and competitive dynamics intensified, pushing some lineages to innovate, migrate, or vanish entirely. Predators had to cover greater distances to locate prey, sometimes venturing into previously uninhabitable zones, while prey developed new avoidance strategies or relied on communal refuge. The narrowing of ecological space created high-stakes encounters, transforming everyday survival into an evolutionary crucible. Species that adapted quickly survived. Those that could not disappeared, leaving behind a record of selective pressure etched into both behavior and population structure. After millions of years of stability, the massive reef structures began to crumble under chemical imbalances and lowered productivity. Growth rates of bryozoans, stromatoporoids, and corals declined, and sections of reef collapsed under their own weight or erosion, leaving skeletal frameworks as silent monuments to former abundance. These remnants became islands for surviving organisms, offering shelter and feeding opportunities. Yet their isolation intensified competition and restricted gene flow. In places where reefs persisted, the survivors' adaptations laid the groundwork for the next wave of ecological expansion, hinting at the resilience that life would draw upon in the face of future crises. Global cooling acted as a selective filter, removing dominant species while leaving space for less conspicuous, more adaptable organisms to flourish. Opportunistic grazers, small scavengers, and burrowers thrived in the surviving habitats, exploiting resources overlooked by larger, specialized species. These survivors became the foundation for future ecological networks, carrying forward adaptations that would shape evolutionary trajectories for millions of years. Even in scarcity, life demonstrated an ability to reorganize, innovate, and persist, proving that ecological collapse can serve as both a destructor and incubator. Nutrient pulses from seasonal currents continue to sustain fragments of life in marginal habitats, allowing micro-communities to persist and maintain minimal ecosystem function. These small populations were not only refuges but also evolutionary laboratories, where environmental pressures drove novel behaviors, reproductive strategies, and physiological adjustments. The survivors' movements, feeding, and interactions subtly reshaped their surroundings, creating feedback loops that reinforced the persistence of life in an otherwise hostile ocean. In these marginal zones, the seeds of future innovation were sown, silently preparing the stage for life's first ventures onto land. Though the ancient seas were scarred and reduced, the remnants of life carried forward adaptations that would anchor new ecological networks, eventually paving the way for terrestrial expansion. Reef patches still pulsed with activity, currents continued to circulate nutrients, and the interplay of predator and prey persisted in surviving communities. The oceans had been reshaped, stripped of some richness, yet imbued with opportunity, demonstrating that even in the wake of catastrophe, life remained resilient and inventive. From these fractured, struggling waters, evolutionary ingenuity quietly prepared the world for its next profound transformation. The Green March, conquest of the continents. Life emerged cautiously onto damp soils, sending root-like structures into sediments that had never felt the touch of greenery. The first vascular plants, small and unassuming, pushed tiny stems upward, capturing sunlight while drawing moisture and nutrients from the shallow floodplains and stream margins. Their presence transformed the once bare substrata, anchoring soil, slowing erosion, and creating microhabitats that retained water and shelter for other organisms. Even in these humble beginnings, the interplay between plant and substrate set the stage for changes that would ripple across the planet, influencing both the chemistry of the land and the currents of nearby waters. Tiny armored arthropods scuttled across muddy surfaces, their segmented bodies moving methodically over roots, detritus, and puddles left by receding tides. These early soil engineers burrowed, grazed, and fragmented organic matter, aerating the sediment and fostering pockets of nutrient-rich soil where future plant growth would thrive. As they travelled, they dispersed spores and tiny plant fragments, carrying genetic material across newly colonized landscapes. Their movements created subtle but essential feedback, accelerating the establishment of complex soil networks while simultaneously shaping the evolutionary pressures acting on plants and other invertebrates. Primitive vascular plants spread along the edges of streams, creating narrow ribbons of green that clung to the water's margins. Rhiniophytes, mosses, and liverworts extended root-like rhizoids into soft sediments, stabilizing the substrate while gradually capturing mineral particles that had never before been bound in living tissue. Their growth not only restructured the physical environment but also increased organic matter accumulation, fostering chemical reactions that gradually transformed the exposed surfaces into the first recognizable soils. As their tissues decomposed, these pioneer plants contributed to nutrient cycles that linked terrestrial margins with aquatic ecosystems, quietly shaping the conditions for life both on land and in nearby waters. Through the slow accumulation of organic matter, soils began to develop in thickness and complexity. These early soils captured moisture, retained warmth, and facilitated the chemical weathering of rocks, slowly releasing minerals that nourished plants while altering river chemistry. Sediment retention improved as roots intertwined with decaying matter, creating microtopographies that supported small pools, channels, and shaded niches. Within these refuges, invertebrates found shelter, while microorganisms processed nutrients that sustained both plants and soil dwellers. The landscape was no longer static. It had become a living tapestry, structured by the interactions of organisms with the earth itself. Chemical fluctuations in soils and water margins created microenvironments of acidity and salinity that challenged some species while providing opportunities for others. Some plants adapted by developing more rigid tissues or deeper rhizoids to anchor in unstable sediments, while arthropods shifted feeding strategies to exploit emerging niches. Detritivores and early decomposers accelerated nutrient cycling, transforming patches of decay into fertile ground that supported successive plant generations. Isolated pockets of life began to harbour unique genetic diversity, allowing early adaptive radiations that hinted at the explosive potential of terrestrial ecosystems yet to come. Carbon dioxide drawdown increased as plant biomass accumulated, subtly cooling the planet over millennia. As photosynthesis captured atmospheric CO2, weathering of exposed rocks and the formation of soils enhanced carbon sequestration, linking biological activity to global climate patterns. Streams carrying mineral-rich runoff affected coastal chemistry and influenced nearshore marine productivity, showing how terrestrial colonization reverberated across the water-land interface. Even the smallest mosses or scattered rhiniophytes played a role in a planetary-scale transformation, demonstrating that life's terrestrial expansion had consequences far beyond immediate habitats. Between early soils and waterways, chemical exchanges created subtle yet profound effects on coastal ecosystems. Dissolved minerals and nutrients carried from terrestrial habitats into rivers and estuaries, enriched shallow marine zones, supporting blooms of plankton and other microscopic life. These nutrient fluxes linked land and sea in a feedback loop, allowing terrestrial innovation to influence marine productivity and oxygen levels. Seasonal pulses of runoff introduced variability in salinity and sediment load, creating new niches for both aquatic and semi-aquatic species. Even small changes in terrestrial erosion rates had cascading effects on coastal habitats, demonstrating that the Green March was not confined to land, but was already reshaping the planet's interconnected systems. Arthropods continued to exploit and reshape these new landscapes, feeding on plants, fungi, and detritus, while distributing spores and seeds. Their burrows maintained soil aeration, and their grazing stimulated plant growth in a dynamic cycle of cause and effect. Each interaction subtly altered energy flow and nutrient distribution, creating networks that were nascent but increasingly resilient. By moving across the damp terrain, these small pioneers facilitated ecological connectivity between isolated plant patches, establishing patterns of life that would later support larger and more complex organisms. Shallow freshwater margins became arenas of experimentation, where primitive vertebrates tested the boundaries between land and water. Early jawless fish and armored ostracoderms ventured briefly into tidal pools and vegetated shallows, encountering new food sources and novel habitats. These interactions catalyzed adaptations such as stronger fins for maneuvering in shallow waters, enhanced sensory perception, and behaviors that foreshadowed terrestrial exploration. Life on land and near-shore waters became intertwined, with each innovation on one side of the interface prompting evolutionary responses on the other. Even in these modest beginnings, the interplay between life and land revealed a profound dynamic. Organisms were no longer passive occupants, but active engineers of Earth's surface. Plants stabilized sediments, arthropods aerated soils, and decomposers recycled nutrients, collectively transforming barren margins into functioning ecosystems. Each biological innovation influenced environmental conditions, which in turn shaped subsequent evolutionary pathways. The once inert landscapes became living systems capable of growth, adaptation, and resilience, illustrating that terrestrial colonization was as much a geological and chemical revolution as it was a biological one. The stage was set for successive waves of terrestrial conquest, each building upon the foundations laid by these pioneering plants and animals. Soil networks thickened, microhabitats expanded, and carbon flows gradually reshaped the climate. By stabilizing sediments, cycling nutrients, and facilitating interactions between species, the first terrestrial communities quietly prepared the land for more ambitious colonizers. The Green March had begun, subtle yet inexorable, signaling that life's transformative reach was no longer confined to oceans and reefs, but now extended firmly onto the continents. Fish that dreamed of air, shallow waters stretched before them, dotted with emergent vegetation and sunlit puddles where oxygen fluctuated daily. Lobe-finned fishes probed these marginal zones, where currents slowed and sediments accumulated, testing the limits of their aquatic design. Each movement of fin or body sent ripples across mudflats that had never felt the weight of a walking vertebrate. Environmental pressure in these transitional waters demanded innovation. Some individuals expanded rudimentary lungs, while others strengthened limb bones, balancing the challenge of respiration with the promise of mobility beyond liquid confines. Life in these murky margins was tentative yet audacious, as evolutionary possibilities extended far beyond the familiar safety of open water. Fins began to stiffen and articulate, hinting at the skeletal structures that would one day support weight on land. The humerus, radius, and ulna of these pioneering creatures lengthened and strengthened, creating joints capable of subtle flexion and leverage. Muscles and connective tissues followed suit, gradually supporting controlled motion across soft substrates. These modifications were not uniform. Populations across separate waterways experimented independently, producing a patchwork of anatomical innovations. Each adaptation carried trade-offs, as increased mobility occasionally compromised swimming efficiency, revealing the delicate balance that shaped early tetrapod evolution. Gills alone could no longer satisfy their metabolic needs, and rudimentary lungs expanded in tandem with exploratory behavior. Air-filled sacs allowed prolonged excursions into oxygen-poor shallow waters and ephemeral pools, reducing dependence on aquatic respiration. Internal circulation is adapted accordingly, ensuring oxygen delivery during intermittent aerial exposure. These physiological shifts coincided with environmental variability, where seasonal drying of shallow ponds created pressures that favored air-breathing individuals. Lungs were not just organs, they were evolutionary invitations, offering access to niches unclaimed by other aquatic life. Scales and dermal armor adjusted to a dual existence, balancing protection against predators with flexibility for movement over uneven substrates. Armor patterns became lighter near limb joints, allowing articulation without sacrificing defense, while cranial plates redistributed to support both aquatic pressure and terrestrial stability. Sensory canals adapted to detect vibrations both in water and in mud, providing early feedback for locomotion and hunting. Even the smallest modifications reflected experimentation as populations navigated the fine line between aquatic efficiency and terrestrial opportunity. Flooded plains and tidal flats offered both danger and opportunity, pressuring populations to innovate or perish. Predators patrolled the shallows, while vegetation and decaying plant matter created hiding spots and feeding grounds. Sediment thickness varied daily, creating temporary islands, channels, and soft beds that tested balance, coordination, and fin strength. Individuals that mastered these shifting landscapes not only survived, but also shaped the environment, displacing sediments and altering microhabitats for both themselves and other organisms. The interplay between challenge and adaptation accelerated evolutionary experimentation. Neck and cranial modifications allowed better scanning of the environment, enabling the earliest prey detection beyond water. Flattened skulls permitted subtle lateral movements, while the separation of head and pectoral girdle gave early tetrapods a wider field of vision. Eyes adjusted to refraction differences between water and air, providing cues for navigation and predation. These structural refinements were coupled with enhanced musculature, allowing precise control over jaw and fin movements. Each increment of mobility and perception multiplied the organism's capacity to interact with both submerged and exposed terrain. Pairs of limbs moved experimentally, pushing against mud and detritus, testing balance, strength, and the promise of terrestrial mobility. Limbs extended from flexible fins, toes, and digits, forming as small nodules of cartilage before ossification. Exploratory movements were intermittent. One limb would anchor in soft substrate, while another probed forward, producing rudimentary crawling patterns. These behaviors were guided by instinctive feedback loops, with failure providing lessons in balance, substrate interaction, and limb coordination. Over generations, trial and error transformed simple fins into structures capable of supporting partial terrestrial locomotion. Behavioral innovations complemented anatomical change, as these early explorers alternated between suction feeding in water and browsing in shallow margins. Mouthparts adapted to capture prey both underwater and at the water's edge, while sensory cues guided foraging on mudflats, algae mats, and invertebrate colonies. Seasonal floods created temporary corridors linking pools and channels, encouraging movement and exploration, while isolated water bodies nurtured population divergence. The combination of structural, physiological, and behavioral adaptations allowed these pioneers to exploit a diversity of ecological niches unavailable to strictly aquatic species. Small population pockets in isolated waterways fostered rapid diversification, laying the groundwork for multiple evolutionary experiments. Each pond, channel, or tidal flat acted as a laboratory, testing combinations of limb structure, respiration, and behavior. Genetic isolation amplified variations, producing lineages with distinct anatomical solutions for shallow water and semi-terrestrial life. Competition and predation pressures reinforced selective advantages, with only the most adaptable surviving repeated environmental fluctuations. These early evolutionary laboratories hinted at the explosive diversification that would follow in later periods, when descendants fully colonized terrestrial landscapes. Chemical and mineral interactions in these marginal zones also guided adaptation, as nutrient availability shaped growth rates and tissue density. Calcium, phosphorus, and trace metals deposited in sediments contributed to stronger skeletal development, influencing both fin robustness and cranial structures. Seasonal pulses of water and detritus redistributed these minerals, creating subtle spatial gradients in which certain populations thrived and others lagged. Environmental chemistry thus became an invisible sculptor, nudging morphology and physiology in tandem, a quiet but persistent partner in evolution's experiment. From these tentative forays, the blueprint for land vertebrates began to take shape, signaling a future dominated by creatures no longer bound to the oceans. Limb articulation, lungs, cranial mobility, and behavioral experimentation coalesced into a coordinated strategy for life beyond water. Every shallow pool crossed and muddy bank explored added to the repertoire of adaptations, generating diversity that would echo for hundreds of millions of years. The boundary between water and land was no longer an absolute barrier, but a frontier of opportunity, a testing ground where evolution's most daring experiments began to unfold. By embracing both worlds, these pioneering fishes had begun the slow, deliberate journey toward the terrestrial giants that would eventually roam Earth's continents. The Devonian darkening. The once stable shallow seas now simmered with volatility, where shifting currents and erratic storms disturbed every ecosystem they touched. Oxygen levels in the water fluctuated unpredictably, creating pockets of suffocation that forced even the most adaptable lobe-finned fishes to seek refuge or perish. Pools that had nurtured early tetrapod excursions alternated between life-giving and lethal, depending on the capricious rise and fall of tides and seasonal rainfall. Currents carried sediments rich in minerals, yet laden with decaying organic matter, altering water chemistry in ways that challenged respiration, digestion, and sensory perception. Every ripple, every murky swirl of debris reshaped survival strategies, demanding innovation in movement, feeding, and shelter. Placoderms and armored fishes that had once dominated these waters vanished suddenly from sediment records, their abrupt disappearance leaving gaps that reshaped food webs. Fossilized evidence shows not only missing populations, but also skeletal deformities in surviving individuals, reflecting stress, oxygen deficiency, and environmental toxins. In these turbulent conditions, competition intensified, not just among species but within populations, as only the most flexible and opportunistic could navigate the rapid changes. Predators that survived were forced to adapt hunting strategies, extending foraging ranges and exploiting new niches, while prey species altered behavior, morphology, and timing of activity to persist in a precarious world. Coastal and marginal habitats fared no better, where erosion and desiccation carved the landscape into fragmented ponds and channels. Early plants struggled as fluctuating water tables and periodic flooding threatened root stability, while sediments shifted with each storm, burying seedlings or exposing roots to air and sunlight. Yet in some pockets, resilience emerged. Small clusters of primitive vascular plants clung to substrate, stabilizing microhabitats that would serve as cradles for future terrestrial life. These surviving plant networks subtly influenced soil development, moisture retention, and nutrient availability, quietly preparing the land for the next wave of ecological expansion, even amid ongoing catastrophe. Water chemistry became a silent but powerful agent of selection. Nutrient pulses from terrestrial runoff spurred algal blooms, which in turn created hypoxic zones and toxic microenvironments. Shifts in calcium and phosphate concentrations affected skeletal growth and shell strength, altering survival odds for fishes and invertebrates alike. The interplay of chemistry and biology transformed habitats into arenas of high-stakes experimentation, where rapid adaptation or extinction determined the course of evolution. Subtle gradients of mineral concentration became selective landscapes, guiding which lineages would persist, migrate, or disappear. Coral frameworks crumbled under stress, reef structures fractured, and symbiotic organisms lost their delicate balance. The collapse of reefs reverberated through food webs. Filter feeders starved, predator-prey relationships shifted, and microbial mats proliferated in newly opened niches. Even microscopic plankton communities responded, their blooms increasingly irregular, contributing to both oxygen fluctuations and sediment deposition. These cascading disruptions underscore that ecosystem stability is fragile, dependent on interconnected life forms that, once removed or altered, amplify instability across habitats. Behavioral adaptations accelerated in response to the chaos. Lobe-finned fishes tested deeper waters, migrated to sheltered inlets, or explored ephemeral pools with heightened caution. Early tetrapods alternated between brief terrestrial excursions and retreat to deeper refuges, their mobility a lifeline against environmental unpredictability. Feeding strategies diversified. Some species scavenged detritus left by mass mortalities, while others adopted ambush tactics, exploiting gaps created by the sudden loss of competitors. This behavioral plasticity became as crucial as anatomical innovation in determining survival outcomes. Isolation of populations intensified evolutionary experimentation. Pools, channels, and floodplain remnants acted as miniature laboratories where genetic drift, selective pressure, and environmental constraints converged. Populations developed unique solutions to repeated desiccation, predation, and oxygen limitation, giving rise to microendemism and divergent lineages. Variations in limb structure, cranial flexibility, and respiratory adaptations accumulated in different pockets, producing a mosaic of morphological experiments that would influence future vertebrate radiations. These isolated microcosms highlight the balance between vulnerability and opportunity in times of widespread environmental stress. Topography and landscape heterogeneity became critical in mediating survival. Minor elevations, shallow ridges, and buried debris created refuges where water persisted longer and predation pressure was reduced. These subtle variations allowed small populations to endure long enough for evolutionary adjustments to manifest. Over time, Surviving lineages exploited these micro-refuges to expand, gradually recolonizing affected areas once environmental conditions moderated. The unevenness of the terrain, previously incidental, became a decisive factor in shaping species survival and dispersal patterns. Even chemical stressors contributed to evolutionary opportunity. Fluctuating oxygen and nutrient levels, coupled with episodic acidic pulses from decaying vegetation, place selective pressure on gill morphology, respiration rates, and metabolic efficiency. Individuals capable of tolerating low oxygen water or exploiting nutrient-rich sediments gained a survival edge, while others succumbed. These chemical challenges accelerated experimentation with physiological and anatomical traits, fostering innovations that would ripple through successive evolutionary generations. Amid this turmoil, new ecological interactions emerged. Opportunistic species occupied vacated niches, establishing predator-prey relationships and territorial hierarchies that had never existed in these seas. Early benthic invertebrates, algae, and surviving vertebrates interacted in novel ways, reinforcing microfood webs and promoting localized stability. Even as dominant species vanished, ecosystems were not inert. Life actively reorganized, illustrating resilience in the face of planetary-scale disturbance. By the conclusion of this darkened era, the Devonian seas were irrevocably transformed. Dominant lineages had fallen. Survivors bore marks of physiological and ecological stress, and emerging communities hinted at the opportunities forged through adversity. Shallow waters, marginal pools, and disrupted coastal zones became crucibles for experimentation, where adaptations in respiration, locomotion, feeding, and behavior combined to create the next generation of evolutionary possibilities. Life, though tested and wounded, persisted, setting the stage for the lush, carboniferous forests and the terrestrial giants that would eventually inhabit them. The darkening was both an end and a prelude, illustrating that chaos could simultaneously destroy and catalyze, preparing Earth for its next remarkable ecological chapter. Forests of Titans, the Age of Coal. Emerging from the experimental margins of Earth's post-Devonian landscapes, vast wetlands sprawled like green oceans across lowlands and deltas, their waters teeming with life, and their edges thick with the first hints of towering forests. These wetlands, nurtured by consistent rainfall and sluggish drainage, became crucibles for gigantism, where both flora and fauna were able to push the boundaries of size and structural ambition. Mosses, primitive ferns, and the earliest lycopsids carpeted the ground. But soon these low-lying pioneers gave way to vertical giants that would dominate the skyline for millions of years, their shadowed bases hiding a bustling network of invertebrates and amphibians. Towering lycopsids pierced the mist, their heights rivaling modern redwoods, while their shallow roots spread through saturated soils, anchoring entire ecosystems and stabilizing the increasingly complex peatlands that would one day form vast coal seams. Their branching patterns, spiral leaf scars, and spore-bearing cones created microhabitats at every level, supporting countless smaller organisms that fed, reproduced, and nested amid the dense vegetation. These forests were not static. They grew, toppled, and decomposed in cycles that redistributed nutrients and reshaped both soil and hydrology, establishing a rhythm of creation and decay that drove ecosystem innovation. Beneath these giants, labyrinthine channels and stagnant pools hosted amphibians, crustaceans, and early insects whose movements stirred sediments, oxygen levels, and nutrient fluxes alike. Primitive tetrapods navigated shallow waters with deliberate motion, their limbs and lungs still in experimental stages, while crustaceans scuttled along submerged routes, filtering detritus and sustaining localized food webs. Each organism contributed to the circulation of energy and materials, their collective activity transforming the wetlands from passive landscapes into highly interactive systems where life and environment continuously molded one another. Air itself became a potent agent, enriched with unprecedented oxygen concentrations that allowed arthropods to grow to sizes unimaginable in later eras. Dragonfly ancestors, such as Meganeura, extended their wingspans beyond 70 centimeters, patrolling the humid skies with predatory precision, while millipede-like Arthropleura crawled across the forest floor, stretching meters in length as they foraged on detritus and decaying plant matter. Elevated oxygen not only facilitated gigantism, but also increased metabolic capacity, influencing behavior, endurance, and the speed of ecological interactions, creating an environment where scale became both a survival advantage and a defining feature of the Carboniferous biome. Leaves, fronds, and branches intercepted sunlight, channeling energy into dense biomass while driving localized humidity and shaping microclimates beneath the canopy. Evapotranspiration created pockets of warm, saturated air, promoting fungal growth, spore dispersal, and small-scale rainfall cycles within the forests themselves. The interplay between photosynthesis, respiration, and water flux established a delicate balance where light penetration, humidity gradients, and substrate saturation influenced species composition at every vertical and horizontal layer of the forest. Decomposition of fallen vegetation and peat accumulation captured carbon, thickening soils, altering nutrient flows, and contributing to the planet's burgeoning coal deposits. Layers of organic matter accumulated faster than they could oxidize, storing vast quantities of carbon underground and influencing global climate through gradual sequestration. The chemical composition of these soils and peat bogs also created microhabitats, rich in minerals, fostering fungal symbioses and microbial communities that further accelerated nutrient cycling and ecosystem productivity. Predators and herbivores engaged in complex interactions, from massive aerial hunters to amphibians navigating flooded undergrowth with precise locomotion and hunting strategies. Early tetrapods stalked the margins of channels, probing the water for crustaceans and larvae, while predatory insects leveraged wings and mandibles to dominate smaller arthropods. These interactions catalyzed adaptations in sensory organs, locomotion, and defensive strategies, generating a mosaic of evolutionary pressures that rippled through the Carboniferous biota, reinforcing both specialization and resilience. Episodic droughts and fluctuating water levels created tension within these thriving forests, forcing migration, dormancy, or physiological adaptations across species. Swamps occasionally receded, exposing mudflats and concentrated prey, while other zones remained submerged, challenging amphibians and insects to exploit or endure variable conditions. These environmental oscillations promoted diversification, selecting for traits that allowed organisms to survive both inundation and desiccation, and fostering communities capable of responding to sudden climate shifts. Chemical shifts in sediment and water influenced oxygen solubility, microbial activity, and nutrient availability across the wetlands. Periodic influxes of minerals, organic detritus, and dissolved gases altered redox conditions, sometimes triggering local hypoxia or surges of productivity that affected community composition. Such geochemical variability created a dynamic template on which life could experiment, reinforcing the role of environmental feedback in shaping ecological trajectories and evolutionary pathways. Even amid abundance, the subtle fingerprints of future ecological stress appeared, foreshadowing the die-offs and climate-induced transitions that awaited the Carboniferous world. Fires, localized drying, and competition for light and nutrients demonstrated that even these monumental forests were not invulnerable, and that dominance carried inherent risk. These pressures, though intermittent, honed survival strategies and set the stage for the eventual collapse of some communities, leaving behind coal-rich strata and evolutionary lessons encoded in every peat layer. Amidst the colossal flora and fauna, the interactions between decomposers and mineral cycles quietly reshaped the forests from beneath. Fungi, bacteria, and detritivorous arthropods penetrated dense leaf litter and fallen logs, accelerating nutrient recycling and influencing spore dispersal patterns of surrounding plants. Microbial mats in shallow pools modified local chemistry, promoting the precipitation of minerals that reinforced root structures and stabilized waterlogged soils. Even small invertebrates burrowed into peat layers, aerating the substrate and creating microchannels that facilitated gas exchange, water infiltration, and seedling establishment. These subtle, unseen processes underscored that the towering spectacle above relied as much on microscopic engineering as it did on giant trees and enormous insects, weaving an intricate web of dependence that strengthened the Carboniferous ecosystem's resilience. In these monumental forests, life reached a breathtaking apex of size, complexity, and interdependence, yet the balance between flourishing and collapse remained delicate. Towering plants, enormous insects, and sprawling wetlands illustrated the heights of ecological ambition, while the interplay of water, oxygen, and biomass hinted at the fragility underpinning even the most titanic ecosystems. This era, though abundant, whispered of impermanence, preparing the narrative for the eventual decline and transformation of these coal forests under the pressures of drying climates, climatic shifts, and changing landscapes. The hemlock skies, collapse of the Carboniferous world. Once teeming wetlands now shrank beneath the pressure of drying climates and shifting watercourses, their vast pools of stagnant water receding under relentless seasonal swings. What had been labyrinthine networks of rivers and deltas transformed into patchworks of isolated ponds and marshes, exposing peat layers that had accumulated over millions of years to air and sunlight for the first time. In these conditions, delicate plant tissues and submerged roots desiccated, leaving skeletal imprints of forests that had once seemed eternal, while pools that remained bristled with surviving arthropods and amphibians struggling to adapt. The balance between water and land, so finely tuned for life's proliferation, now teetered on the edge of instability, foreshadowing the upheaval that would reshape every corner of the Carboniferous world. Massive continental collisions lifted mountain ranges that diverted rivers and fractured habitats across vast regions, creating barriers that altered climate patterns and moisture distribution. Rain shadows fell across previously lush lowlands, and valleys that had once nurtured coal forests became arid corridors where plant growth faltered. Sediment deposition shifted dramatically as new uplifts redirected flow paths, altering nutrient availability and forcing organisms to contend with novel, fragmented landscapes. The slow, inexorable grinding of continents against one another became an invisible architect of ecological stress, sculpting a terrain that challenged even the most resilient species. Atmospheric oxygen, once abundant and sustaining gigantic insects and thriving amphibians, began to fluctuate with alarming irregularity, subtly altering physiology and behavior across ecosystems. Spiracles and tracheal systems evolved to deliver oxygen to enormous bodies, now faced limitations, curbing maximum insect size and influencing patterns of locomotion and feeding. Vertebrates sensitive to oxygen content experienced similar pressures, and some lineages contracted in range, while others experimented with more efficient respiratory strategies. The unseen chemistry of the air had become a critical force, silently dictating the fate of life across continents. Peatlands, the repositories of ancient carbon, slowly decomposed as water tables fell and fungal activity accelerated, releasing trapped carbon into the atmosphere and contributing to climate variability. Layers of accumulated vegetation, once destined for coal formation, now decayed in open air, leaving only faint impressions of their former abundance. Microbial and invertebrate decomposers penetrated these mats, hastening nutrient turnover and subtly modifying soil chemistry, yet the balance favored release over sequestration, signaling a profound environmental shift. The collapse of these carbon reservoirs was not merely a loss of material, but a recalibration of the planetary carbon cycle itself. Insects that once roamed with ease now faced shrinking niches, forcing adaptations in flight, size, and behavior. Giant dragonflies and mayflies confronted increasingly patchy habitats, and their predation strategies required modification as prey became dispersed and ephemeral. Morphological plasticity became a survival advantage, with some lineages exhibiting reduced size to navigate narrow vegetative corridors or altered mouthparts to exploit diminishing resources. In the skies and canopies of these dying forests, survival depended on flexibility, ingenuity, and the ability to respond to forces far beyond any individual organism's control. Tree canopies, once uninterrupted, are fragmented under climatic stress, creating corridors for sunlight but exposing seedlings to desiccation and predation. The architecture of forests, carefully built over millennia, now faltered, and fallen logs littered the once continuous understory. Seedling recruitment became precarious, and reproductive cycles shifted in response to the patchwork of surviving niches. Ecological interactions, from competition for light to herbivory pressure, intensified within these fragmented spaces, revealing that even the mightiest arboreal giants were vulnerable to systemic change. Fire became an increasingly common sculptor of the landscape, consuming swaths of undergrowth and leaving charred remnants of towering lycopsids. Lightning strikes, lightning-driven wildfires, and seasonal droughts combined to transform large areas into open, nutrient-rich ground, temporarily favouring opportunistic plant species. The smoke and heat not only removed biomass, but also altered local atmospheric chemistry, influencing humidity, seed dispersal, and microclimates. Through these infernos, ecological reset mechanisms operated at scales previously unseen, leaving behind a mosaic of destruction and potential regeneration. Coastal plains and delta systems eroded as sea levels fluctuated, altering sediment deposition and nutrient availability for both plants and aquatic organisms. Channels that had funneled fresh water to wetlands became unstable or disappeared entirely, leaving some areas parched and others flooded episodically. Saltwater intrusion reshaped estuarine ecosystems, challenging freshwater flora and fauna with rapidly changing osmotic conditions. The interaction of tectonics, climate, and hydrology produced landscapes in constant flux, where survival required both adaptability and sheer luck. In the wake of mass extinctions, early amniotes and other resilient vertebrates began exploring new ecological opportunities left vacant by extinctions. These pioneering species expanded into niches freed by the decline of amphibian-dominated communities, exploiting dry floodplains, partially desiccated wetlands, and open forest edges. Limb morphology, respiratory efficiency, and reproductive strategies diversified rapidly, laying the groundwork for future dominance of terrestrial vertebrates. Life pressed forward, quietly inventing strategies that would define the next evolutionary epoch despite the fragility surrounding them. Geochemical records reveal pulses of carbon and sulfur shifts, marking episodes of environmental stress visible across the globe. Isotopic analyses capture spikes in atmospheric CO2 associated with peat oxidation and volcanic activity, while sulfur signatures indicate marine anoxia events and altered nutrient flux. These chemical fingerprints confirm that the Carboniferous collapse was not a localized phenomenon, but a planet-wide recalibration linking terrestrial decline to oceanic and atmospheric instability. The narrative of forest demise is thus intertwined with deep Earth processes, emphasizing that global forces and local ecosystems are inseparably connected. From the remnants of these forests, life that endured quietly readied the stage for the next era, where reptilian lineages would rise to dominance. The immense biomass, nutrient deposits, and surviving habitats provided a foundation for diversification and adaptation, even as the Carboniferous giants faded into memory. Ecological opportunity and evolutionary experimentation flourished in the aftermath of destruction, proving that collapse, though devastating, is never final. In the skeletal remains of lycopsid fortresses and drained peatlands, the seeds of the Mesozoic future were already taking root, awaiting the creatures that would eventually claim the land as their own. Reptiles take the stage. Out of the shadows cast by desiccated wetlands, the earliest reptiles seized the land with a quiet audacity previously unseen in vertebrate history. The remnants of Carboniferous forests and drained peatlands offered both challenge and opportunity, shaping ecosystems that demanded resilience and innovation. Unlike amphibians bound to moist habitats, these pioneering amniotes could exploit newly exposed soils, floodplains, and forest margins using small body size, metabolic efficiency, and subtle locomotor flexibility to move across substrates that had once been forbidding. Their expansion marked the first sustained vertebrate mastery of dry terrestrial environments. Their amniotic eggs, encapsulated in protective membranes, freed reproduction from the vagaries of water, allowing colonization of arid floodplains, upland terraces, and sporadic forest patches. Each embryo was shielded from desiccation and predation, a compact evolutionary package that enabled long-distance dispersal and habitat occupation previously impossible for amphibians. This reproductive innovation set a precedent that would define vertebrate success for hundreds of millions of years, providing ecological leverage, allowing population stability, and opening corridors for the exploration of diverse niches that stretched from river margins to semi-arid plains. Limbs strengthened with reinforced joints and claws, adapted for traction, allowed these reptiles to traverse substrates that had been impassable for their amphibian predecessors. Articulated digits, enhanced musculature, and flexible axial skeletons provided both speed and endurance, enabling active pursuit of prey and rapid escape from predators. Locomotor versatility permitted exploitation of complex terrain, from fallen logs to muddy banks and sandy flats, while their ability to climb, burrow, or perch subtly extended their ecological reach. By moving with efficiency and control, these early reptiles began shaping ecosystems through behavior as much as physiology. As sunlight warmed open landscapes, thermoregulation became an essential strategy, influencing daily activity, foraging, and survival. Some species basked to accelerate digestion and muscle performance, while others adopted nocturnal or crepuscular habits to avoid overheating or predation. Behavioral flexibility complemented anatomical innovation, permitting reptiles to exploit a wider thermal envelope than amphibians could tolerate. By regulating body temperature with postural adjustment, pigmentation changes, and microhabitat selection, they gained access to habitats that fluctuated dramatically in moisture, exposure, and temperature, providing a competitive advantage that reinforced the trend toward terrestrial dominance. Jaw mechanics evolved to handle diverse diets, from insects and small vertebrates to opportunistic scavenging, expanding the ecological versatility of these pioneering reptiles. Stronger adductor muscles, specialized tooth arrangements, and refined bite mechanics enabled precise capture and processing of prey, while some lineages developed slicing, crushing, or shearing capabilities. This dietary flexibility allowed species to occupy multiple trophic levels simultaneously, supporting population growth and promoting diversification. By exploiting previously inaccessible food sources, early reptiles reshaped energy flows, created selective pressures for other organisms, and established the foundation for increasingly complex terrestrial food webs. Skull and vertebral modifications, including interlocking vertebrae, reinforced rib cages, and expanded cranial capacity, supported more active locomotion, efficient respiration, and greater behavioral sophistication. These skeletal adaptations permitted sustained movement across varied terrain, while stabilizing the body during rapid bursts of activity, climbing, or digging. Structural improvements in vertebrae and ribs also facilitated expansion of lung capacity, increasing aerobic performance and allowing reptiles to inhabit drier, oxygen-variable environments. The interplay of skeletal robustness, respiratory efficiency, and postural control was crucial in transforming the landscape into a habitat that reptiles could actively manipulate and dominate. Early diversification saw lineages specialize into distinct niches. Some adapted for rapid bursts across open terrain, others for stealthy hunting along river margins and forest edges, and yet others for burrowing or perching. Each strategy involved subtle combinations of limb morphology, sensory enhancement, and behavioral innovation, creating a mosaic of ecological roles that reduced competition and promoted coexistence. Habitat partitioning encouraged adaptive radiation, and geographic variation further accelerated divergence. Through these mechanisms, reptiles established resilient populations in regions that amphibians could not permanently occupy, gradually asserting dominance over terrestrial ecosystems and altering evolutionary trajectories. Predation pressures intensified as amphibians and synapsid relatives contended with reptiles' emerging dominance, creating novel evolutionary feedback loops. Small carnivorous reptiles hunted insects and juvenile vertebrates, while larger species occasionally preyed upon amphibians weakened by desiccation or disease. Defensive traits in prey, including burrowing, nocturnality, and armor-like dermal adaptations, evolved in response, establishing early arms races that drove anatomical and behavioral specialization. Through competition, predation, and exploitation of new habitats, reptiles catalyzed ecological restructuring, reinforcing their ascendancy while simultaneously shaping the evolutionary potential of coexisting lineages across emerging terrestrial landscapes. Environmental instability continued, but reptiles' resilience to drier climates, fluctuating water availability, and fragmented habitats allowed them to thrive where other lineages contracted or disappeared. Seasonal droughts, temperature swings, and occasional flooding created selective pressures that favored physiological efficiency behavioral flexibility, and dispersal capacity. By exploiting microhabitats, using burrows, or adjusting activity patterns, these creatures maintained viable populations and expanded into marginal environments. Their ability to buffer against environmental variability provided both survival advantage and evolutionary momentum, enabling reptiles to act as ecological engineers whose presence influenced soil stability, plant growth, and the broader structure of terrestrial ecosystems. Geochemical and paleobotanical evidence reveals how early reptilian expansion reshaped soil composition, nutrient cycling, and terrestrial food webs. By burrowing, trampling, and feeding, reptiles altered microbial communities, enhanced aeration, and redistributed organic matter, indirectly influencing plant community structure. Their interactions with insects, small amphibians, and early plants contributed to the emergence of layered ecosystems, with primary producers, consumers, and decomposers forming increasingly integrated networks. These ecosystem engineering effects amplified ecological complexity, ensuring that reptiles did not merely occupy existing niches, but actively created conditions for further diversification, laying the groundwork for the Mesozoic proliferation of terrestrial life. By claiming these landscapes, reptiles set the stage for the Mesozoic, demonstrating that the ability to innovate physiologically and behaviorally could transform ecosystems on a planetary scale. Their pioneering amniotic strategies, skeletal modifications, and behavioral versatility established a durable framework for vertebrate expansion. As amphibians receded, deserts, floodplains, and forest edges became arenas of reptilian experimentation, where new feeding strategies, reproductive modes, and social interactions emerged. The early conquest of land was not sudden but deliberate, cumulative, and adaptive, illustrating that the Mesozoic dominance of reptiles began as a quiet, persistent reshaping of Earth's terrestrial canvas. The day life almost ended. As volcanic eruptions intensified, ash and aerosols choked the skies, dimming sunlight and cooling the planet with relentless efficiency. Forests blackened under the weight of falling cinders, and shallow seas were darkened by particulate clouds that blocked photosynthesis. Atmospheric turbulence disrupted rainfall patterns, creating sudden droughts across coastal plains, while inland wetlands stagnated. The delicate balance that had allowed ecosystems to flourish for millions of years was shattered almost overnight. Life's complexity, once expanding with the quiet persistence of evolution, now confronted the brutal reality of planetary upheaval, where only adaptability could determine survival. Oceans stagnated under blankets of anoxic water, suffocating species that had once thrived in vibrant reef and coastal systems. Coral structures collapsed, filter-feeding invertebrates perished, and plankton blooms, once engines of oxygenation, turned deadly under chemical stress. Predatory fish found their hunting grounds empty, and scavenger communities declined as organic matter accumulated faster than it could be processed. Across continental shelves, the collapse of food webs created ecological voids, leaving niches unoccupied and further destabilizing marine communities. Survival was no longer about competition, but about endurance in a fundamentally altered environment. Rivers and floodplains, stripped of vegetation, became channels of sediment and chemical flux, eroding habitats that had sustained amphibians and early reptiles alike. Water chemistry shifted as runoff carried volcanic ash and metals downstream, contaminating aquatic environments. Shallow pools and ephemeral ponds dried rapidly, leaving stranded animals vulnerable to predation and heat stress. Microclimates that had once provided stability vanished, forcing mobile species to traverse treacherous terrain or perish. Even resilient amphibians, with their dual reliance on water and land, faced unprecedented pressure as their breeding and feeding grounds disappeared in the span of decades. Temperature swings and acidifying waters fragmented communities, creating islands of survival amidst landscapes of collapse. Shallow seas experienced cycles of warming and cooling so rapid that organisms could not adapt physiologically, while soils alternated between desiccation and flooding. Symbiotic relationships broke down, leaving plants unable to stabilize sediments and animals deprived of shelter or food. These environmental oscillations selected for lineages capable of tolerating extremes, favoring small body size, rapid reproduction, and flexible behavior. Across continents and oceans, extinction pressure sculpted the biological landscape with precision, erasing forms incapable of adapting in time. During periods of imbalance, predatory pressures shifted abruptly as opportunistic species exploited weakened populations, redefining ecological hierarchies in real time. Scavengers capitalized on sudden die-offs, while survivors expanded into territories previously dominated by more vulnerable species. Feeding strategies evolved under pressure, as carnivores modified hunting behaviors and herbivores adjusted diets to scarce vegetation. Evolutionary experimentation accelerated, but with survival contingent on both skill and chance. In these turbulent interactions, the normally slow rhythms of natural selection were compressed into catastrophic immediacy, producing stark winners and the majority of losers in a single evolutionary heartbeat. Microhabitats, from caves to burrows, became refuges where resilience and behavioral flexibility determined who would endure. Early reptiles and amphibians that could shelter underground or in dense vegetation avoided temperature extremes and predation. Soil burrows buffered against chemical contamination and rapid desiccation, while hollow logs and crevices provided temporary havens for insects and smaller vertebrates. Even within these confined spaces, resource scarcity created intense competition, favoring generalist diets and opportunistic behaviors. Survival became a test of ingenuity, a delicate balance between exploiting available resources and avoiding the hazards of an increasingly hostile world. Reproductive strategies were tested to the limit, with oviparous lineages showing a marginal advantage in surviving isolated, variable environments. Eggs deposited in protected microhabitats were more likely to reach maturity than those exposed to fluctuating moisture, temperature, or chemical stress. Species that could reproduce quickly and disperse broadly gained a foothold, while others faced local extinction. The selective pressures reshaped evolutionary trajectories, favoring traits that combined resilience, efficiency, and flexibility. Even as the planet reeled, the genetic groundwork for future radiations was laid in the survivors, quietly preparing the canvas for the Mesozoic recovery. Fossil evidence of mass mortality layers, bite marks, and sudden burial events reveals the episodic intensity of the global crisis. Sedimentary deposits show repeated pulses of volcanic ash, black shale, and disrupted strata, documenting die-offs in both marine and terrestrial settings. Anoxic events left stark chemical signatures, while articulated skeletons indicate sudden death rather than gradual decline. These geological and biological records confirm that extinction occurred in multiple waves, driven by complex interactions between volcanism, climate instability, and ocean chemistry. Life's fragility was laid bare, but so too was the resilience of those lineages capable of weathering catastrophe. Geochemical anomalies, including spikes in carbon isotopes and mercury, document volcanic and tectonic forces that reshaped Earth's chemistry. Massive releases of CO2 and other greenhouse gases drove rapid warming, while mercury deposits indicate repeated episodes of intense volcanism. Oxygen fluctuations in sediments reveal widespread anoxia, and trace element shifts illuminate the destabilization of nutrient cycles. These measurements provide a precise record of planetary upheaval, illustrating how environmental stresses combine to push ecosystems beyond their tipping points. The scale and rapidity of these events ensured that extinction was both selective and sweeping, redefining the biological landscape. In the aftermath, surviving reptiles and select amphibians became architects of nascent ecosystems, slowly recolonizing niches left vacant by extinction. Opportunistic lineages spread into newly available floodplains, coastal margins, and inland waters, establishing populations that could exploit post-catastrophe abundance. Interactions between surviving species were reshaped, creating simplified yet dynamic food webs. Early terrestrial plants that endured alongside animals stabilized soils and facilitated nutrient cycling, enabling gradual ecosystem recovery. This period of fragile renewal demonstrates that even near total devastation cannot halt life's drive to adapt, innovate, and reclaim opportunity from catastrophe. With devastation behind them, a restructured world emerged, where evolutionary potential remained vast, and the foundations for Mesozoic ecosystems quietly took shape. Reptiles, with their physiological and behavioral innovations, gained prominence, while amphibians and other survivors occupied increasingly specialized niches. New ecological architectures emerged, simpler yet poised for expansion, as evolutionary experimentation resumed in the spaces left by extinction. Life, tested to its limits, retained the capacity to innovate, and the survivors of this global crisis became the architects of a future where the planet could once again host abundant and diverse ecosystems. Catastrophe had reset the stage, but it had not ended the story. The Great Rebuilding, a planet learning to breathe again. Smoke-scarred plains began to shimmer with tentative shoots, their roots threading through ash and silt, claiming spaces where life had almost vanished. The first green tendrils pushed upward in cautious defiance, stabilizing soils and trapping moisture in a landscape still raw from the Permian catastrophe. Every sprout represented both a fragile experiment and a harbinger of ecological restoration, gradually knitting fractured terrains into living networks. Even in the absence of dominant species, the land hummed with latent potential, signaling that evolutionary momentum could resume and that new ecosystems would slowly reclaim the planet. Beneath still waters, the survivors stirred, probing empty niches with cautious movements that hinted at a cautious resurgence. Fishes and invertebrates that had endured the global collapse explored previously vacant reef patches and shallow lagoons, reshaping food webs and nutrient flows. Microbial mats and early algae flourished in tandem, stabilizing sediments while fostering oxygenation. This hidden world of activity laid the foundation for more complex interactions, as each recovered population subtly influenced the chemical and structural dynamics of its surroundings. Life's resilience revealed itself not through spectacle, but through these patient, persistent rhythms. The air itself seemed to thicken with potential as oxygen levels climbed imperceptibly, giving strength to creatures that had endured the darkest days. Surviving reptiles and amphibians gained improved metabolic efficiency, while smaller vertebrates experimented with locomotion, feeding, and reproductive strategies in the newly oxygen-rich habitats. Even slight shifts in atmospheric composition rippled through the recovering biosphere, affecting growth rates, activity patterns, and interspecies interactions. Each breath carried a hint of renewal, reminding the world that recovery depended not just on surviving life, but on the gradual restoration of environmental conditions capable of sustaining diversity. Hidden among fractured landscapes, pockets of shelter nurtured fragile communities, each microcosm a crucible for adaptation. Shallow floodplains, isolated river bends, and temporary wetlands offered niches where species could rebound with limited competition. Within these refuges, variations in behavior, morphology, and life cycle timing emerged, accelerating evolutionary experimentation. Fragmentation promoted diversification even as it imposed constraints, creating a patchwork of specialized populations. These localized bursts of activity collectively contributed to the broader planetary recovery, demonstrating that the spatial complexity of habitats was as critical to the resurgence of life as the survival of individual lineages. Predators re-emerged, calculated and deliberate, testing both prey and environment without triggering collapse in the fragile new order. Insects, amphibians, and early reptiles resumed hunting and foraging, establishing new trophic hierarchies that reinforced ecosystem structure. Bite marks, traces of predation, and foraging scars became evidence of interaction, showing that life was cautiously reconstructing the dynamic networks lost in the mass extinction. These behavioral experiments were constrained by the scarcity of prey and spatial limitations, yet they provided critical feedback loops, ensuring that both predators and their food sources co-evolved in ways that promoted gradual stability. Greens and ferns unfolded across floodplains and riverbanks, capturing light and cycling nutrients into soils that had long lain barren. Photosynthetic organisms generated the first sustained organic matter inputs in many landscapes, feeding decomposers and stabilizing soil chemistry. As root systems deepened, they retained moisture, reduced erosion, and created microhabitats for arthropods, amphibians, and early reptiles. The re-establishment of vegetation drove local climate moderation, influencing humidity, temperature, and seasonal water availability. These botanical engineers quietly orchestrated the groundwork for increasingly complex food webs, demonstrating that plant life could drive ecosystem recovery as much as animal persistence. Ephemeral pools mirrored the skies, providing arenas where the first tentative experiments in behavior and reproduction unfolded. Amphibians and small reptiles returned to water bodies for breeding, navigating unpredictable hydro periods and fluctuating resource availability. Eggs, larvae, and juveniles faced selective pressures that promoted survival strategies such as burrowing, clutch dispersion, and aquatic foraging efficiency. These microecosystems acted as testing grounds, revealing how life could adapt under constraint and ensuring that only the most resilient and flexible lineages persisted. Across these temporary havens, the future blueprint for terrestrial and aquatic interdependence began to emerge. Scattered islands of habitat formed amidst shifting sediments, isolating populations and fostering bursts of morphological novelty. Geographic separation accelerated divergence as small groups of reptiles, amphibians, and invertebrates adapted to unique microenvironments. Variation in limb structure, skin texture, and metabolic function reflected rapid adaptation to local challenges. Even limited gene flow between islands introduced opportunities for hybridization, enhancing diversity. This spatial mosaic of evolving populations exemplified how isolation combined with environmental pressure could catalyze innovation, preparing the stage for more complex Mesozoic ecosystems that would eventually dominate terrestrial landscapes. Chemical traces in the rock whispered of recovery, mapping oxygenation, carbon fluxes, and nutrient returns across oceans and estuaries. Geochemical proxies revealed the gradual normalization of atmospheric and oceanic conditions, documenting the return of habitable environments. Recovered biogeochemical cycles supported plankton blooms, facilitated nutrient recycling, and stabilized oxygen availability, indirectly shaping the evolution of vertebrate and invertebrate communities. These chemical signals acted as silent indicators of planetary healing, providing a scaffold upon which both flora and fauna could expand. They underscored that ecosystem recovery is inseparable from the geochemical context that sustains life. Interactions resumed, intricate and precarious, as species cautiously occupied their new roles in the recovering ecosystems. Herbivores began grazing on emerging vegetation, predators resumed hunting, and decomposers efficiently processed detritus, creating energy flows that supported renewed trophic networks. Mutualistic relationships developed between plants and arthropods, while competition refined behavioral strategies and niche specialization. These interactions, though subtle compared with the exuberance of pre-extinction communities, ensured functional stability. Life was once again experimenting with complexity, proving that even after catastrophic loss, ecological principles could be restored incrementally, guided by both necessity and opportunity. Landscapes hinted at a fragile prosperity, signaling the eventual rise of ecosystems poised to host the next era of vertebrate dominance. Forests began to form in sheltered valleys, river networks stabilized, and faunal communities gained density and specialization. Early reptiles, amphibians, and arthropods exploited these recovering habitats, reinforcing ecological architecture through feedback loops of predation, reproduction, and resource use. The planet, once scarred and nearly emptied of life, now showcased a tentative but tangible resilience. From this delicate renewal, the evolutionary stage was set for the flourishing of Mesozoic ecosystems, where innovation and diversity would again reign supreme. Rise of the archosaurs. A shimmer of movement across drying mudflats revealed tiny predators testing the edges of survival. These early archosaurs navigated fragile soils and receding waters, their limbs and tails adapted for swift movement and sudden bursts of power. Subtle shifts in musculature and posture allowed them to exploit niches abandoned by less resilient amphibians. With each careful step, they redefined the rules of terrestrial competition, their presence signaling the dawn of a lineage destined to dominate swamps, forests, and floodplains alike, quietly rewriting the architecture of life on land. Jagged shadows stretched across riverbanks as reptiles explored unclaimed niches with cautious precision. Social hierarchies and territorial instincts began to emerge, with displays of dominance shaping reproductive success. These pioneering reptiles refined balance, coordination, and sensory acuity, giving them advantages in hunting and evasion. The interplay of environment and behavior created feedback loops of innovation, those best adapted to shifting rivers, seasonal droughts, and predator-prey dynamics thrived, slowly carving a blueprint for the archosaur success story that would come to define the Mesozoic terrestrial landscape. Fossilized skeletons whispered of muscle leverage and jaw power that would define future dominion. The elongation of limbs and reinforcement of vertebrae allowed rapid acceleration and agile turning, while cranial structures evolved to accommodate increasingly diverse diets. Some experimented with ambush strategies along floodplain margins, while others became endurance hunters. Each adaptation reflected selective pressures in a recovering world, and incremental innovations compounded over generations. The very bones of these creatures told a story of evolutionary trial and triumph, capturing the delicate balance between predation, locomotion, and environmental opportunity. Razor-edged teeth and curved claws hinted at evolutionary strategies honed under environmental strain. Carnivorous archosaurs experimented with prey capture across multiple scales, from insects and small amphibians to larger vertebrates navigating unstable floodplains. Parallel developments in digestion, jaw articulation, and forelimb dexterity increased efficiency, while environmental pressures encouraged subtle differentiation between lineages. These innovations foreshadowed future forms capable of dominating entire ecosystems. In each territory claimed, archosaurs refined their abilities, not merely surviving, but shaping the landscape through predation, competition, and interaction with newly forming terrestrial communities. Droughts and seasonal storms sculpted not only the land, but the very forms that inhabited it. Fluctuating water tables and unpredictable rainfall created selection pressures for physiological resilience. Efficient kidneys, flexible respiration, and locomotor endurance became critical. Vegetation patterns dictated cover and ambush opportunities, influencing hunting behaviors. Archosaurs that could traverse exposed plains or endure harsh microclimates gained reproductive and survival advantages. These episodic challenges acted as evolutionary crucibles, ensuring that the lineages which endured were not simply survivors, but architects of a new ecological order, laying the foundations for Mesozoic dominance. Isolated groves became crucibles, where subtle anatomical innovations were amplified across generations. Within these pockets of stability, archosaurs experimented with reproductive strategies, clutch sizes, and nesting behaviors. Competition for limited resources reinforced selective pressures on growth rate, body size, and sensory acuity. Predatory interactions with amphibians and smaller reptiles fostered diversification in hunting methods and locomotion. These evolutionary experiments, repeated and refined across isolated populations, created morphological and behavioral variations that would define crocline and birdline reptiles, setting the stage for their eventual proliferation across global habitats. Amphibians' retreat opened corridors for archosaurs to experiment with movement, hunting, and nesting. As swampy lowlands and dense wetlands lost diversity, new terrestrial corridors became available. Archosaurs explored these niches with iterative trial and error, testing limb articulation, jaw strength, and social behaviors. Such experimentation accelerated evolutionary divergence, giving rise to specialized forms adapted to floodplains, forest edges, and open plains. The decline of amphibian dominance allowed archosaurs to push boundaries of size, mobility, and predation efficiency, transforming the ecological landscape while simultaneously shaping the selective pressures for their own continued innovation. Embedded in ancient soils, chemical signatures recorded oxygen fluctuations that pressured adaptation in unexpected ways. Periodic hypoxia and temperature swings challenged metabolism, encouraging respiratory efficiency and behavioral flexibility. Archosaurs capable of sustained activity in oxygen-poor or thermally variable environments gained an edge in colonizing exposed habitats. Soil nutrient cycles, altered by plant recolonization and early microbial activity, influenced prey abundance and habitat suitability. These interconnected abiotic and biotic factors ensured that evolutionary success was contingent on both adaptability and ingenuity, driving the refinement of traits that would characterize the dominant reptiles of the Mesozoic. Territorial displays and foraging patterns began to shape the first social hierarchies among these reptiles. Courtship, resource defense, and cooperative behaviors emerged gradually, allowing individuals to monopolize hunting grounds or nesting sites. Such interactions promoted communication, memory, and strategic planning, laying the groundwork for complex behaviors observed in later archosaurs. These early social structures, though primitive, were crucial in stabilizing populations and enabling lineage persistence. Success became as much a product of behavioral acumen as physiological strength, demonstrating that ecological mastery required the integration of body, brain, and environment. Floodplain corridors became evolutionary highways, guiding the expansion of lineages destined to dominate. Seasonal rivers and vegetated banks provided connective pathways between resource-rich regions, allowing dispersal and genetic exchange. Populations that traversed these corridors rapidly diversified, encountering new prey types, competitors, and microclimates. Over time, selective pressures in these corridors reinforced traits like endurance, speed, and cognitive problem-solving, accelerating divergence between crocline and birdline archosaurs. These migrations set the stage for global radiation, foreshadowing the eventual dominance of archosaurs across continents, ecosystems, and climatic zones during the Triassic and beyond. At the margins of ancient forests, archosaurs demonstrated the incremental mastery of the recovering world. Here, adaptations in limb articulation, sensory acuity, and metabolic efficiency were tested against predation, resource scarcity, and seasonal variation. Every success and failure shaped evolutionary trajectories, reinforcing the subtle but cumulative nature of archosaur innovation. Their careful exploration of diverse microhabitats, combined with physiological resilience and behavioral plasticity, ensured that surviving lineages were poised for expansion. In these margins, the blueprint of Mesozoic supremacy began to crystallize, illustrating how life exploits opportunity even in fragile, post-extinction landscapes. Dawn of the dinosaurs. Slender shadows weaved between ancient tree trunks, testing every crack and crevice for survival. These first small dinosaurs navigated a world still dominated by archosaur competitors, learning to exploit gaps in vegetation and microhabitats overlooked by larger predators. Their bodies were lightweight, limbs articulated for speed and maneuverability, and tails served as balancing rudders. Each movement was honed through trial and error, shaping hunting strategies and predator avoidance. In these forests and floodplains, life's blueprint was being refined, setting the stage for the evolutionary explosion that would define the Mesozoic. Claws struck at the moist soil, uncovering insects and small vertebrates that would fuel the next generation. Dietary specialization was emerging, with opportunistic feeding granting advantages over less adaptable contemporaries. Jaw structure and dentition began to reflect subtle shifts toward carnivory and omnivory, while cranial flexibility enhanced processing of diverse prey. Even within this marginal niche, competition was fierce, driving incremental improvements in sensory acuity and coordination. Each foraging expedition offered lessons in efficiency, endurance, and survival, demonstrating that small size could be a formidable advantage in post-extinction ecosystems. Wings of dragonfly ancestors shimmered overhead, forcing fledgling hunters to adapt instant reflexes. The aerial environment was alive with swarms of insects, shaping the hunting techniques of emerging dinosaur lineages. Vision and rapid response became critical traits, while limb and tail coordination evolved to exploit these abundant but elusive food sources. Swampy habitats, river margins, and fern understories offered both refuge and challenge, encouraging behavioral plasticity. Over generations, natural selection favored those capable of reading movement, timing strikes, and avoiding predation. embedding these skills into the lineage that would eventually dominate terrestrial ecosystems. River currents reshaped the mudbanks daily, creating constantly changing pathways and hunting grounds. Seasonal flooding and sediment deposition altered habitats, exposing new feeding opportunities while erasing familiar refuges. Small dinosaurs adapted to these shifting landscapes through agility, exploratory behaviors, and keen spatial awareness. Limb morphology and foot structure reflected repeated adaptations to slippery mud, dense vegetation, and irregular terrain. By negotiating these unstable environments, they honed not only survival skills, but also ecological intelligence, learning patterns that would inform dispersal, territoriality, and reproductive site selection for generations to come. Footprints, etched into silt, reveal fleeting alliances and rivalries among the earliest small dinosaurs. Evidence of grouping behavior, territorial boundaries, and predator evasion appears in trackways preserved for hundreds of millions of years. These imprints suggest dynamic social interactions, cooperative strategies, and early attempts at parental care or communal defense. Patterns in stride length, spacing, and trail overlap hint at communication mechanisms, whether visual, auditory, or chemical. In these ancient records, the story of emerging dominance and ecological experimentation is preserved, demonstrating that even diminutive creatures could impact population structures and influence survival across vast floodplains and forested regions. Sunlight fractured through conifer canopies, highlighting patterns of motion that enhanced predator efficiency. Light and shadow created natural camouflage for both prey and predator, demanding heightened perception and cognitive mapping. The interplay of illumination, vegetation, and movement shaped hunting strategies, with successful individuals exploiting temporal and spatial advantages. Eyes positioned for binocular depth perception and neck mobility for scanning became selective traits, while behavioral routines incorporated patterns of light, cover, and motion. These subtle environmental pressures fostered innovations that would later allow dinosaurs to occupy niches ranging from underbrush predators to apex hunters. A sudden predator's roar sent tremors across the floodplain, reinforcing the need for agility and vigilance. Survival demanded instantaneous reaction to threats, selecting for rapid reflexes, burst speed, and acute auditory perception. Smaller dinosaurs adapted not only to predation by larger archosaurs, but also to the unpredictability of natural hazards, such as sudden floods, fallen trees, or volcanic activity. Risk assessment, memory of escape routes, and coordinated movement became embedded behavioral traits. Each encounter with danger was an evolutionary lesson, ensuring that only the swift, alert, and adaptable individuals passed on genes shaping future generations. Nest sites concealed in fern tangles illustrate early attempts at reproductive protection and survival strategy. Egg-laying behavior, site selection, and even rudimentary nesting construction reflect selective pressures on reproductive success. Proximity to food, predator avoidance, and microclimate regulation influence survival rates, while social behaviors may have included guarding, site fidelity, or even communal nesting. Early reproductive innovation ensured that lineages persisted despite environmental instability, linking behavioral sophistication with evolutionary resilience. In these small actions, dinosaurs began to exercise control over generational survival, laying the foundation for the reproductive versatility that would later characterize entire clades. The hum of swamp insects and distant amphibian calls formed a sensory backdrop that shaped behavior. Auditory cues became critical for detecting prey, avoiding predators, and navigating complex environments. Vocalization, rustling, and movement all contributed to a multisensory landscape demanding constant attention. Behavioral plasticity, attention allocation, and rapid decision-making were tested continuously, favoring individuals capable of processing multiple stimuli simultaneously. In this oral ecosystem, sensory integration and learned responses drove innovation in feeding, evasion, and interaction, embedding traits that would define dinosaur cognition and ecological mastery for millions of years. Seasonal shifts forced populations to adjust their diet, movement, and timing, selecting for adaptable traits. Fluctuating temperatures, rainfall, and food availability created cycles of scarcity and abundance, shaping migration, breeding, and growth strategies. Flexible metabolism, opportunistic feeding, and hibernation-like behaviors emerged in response, allowing survival across fluctuating conditions. These pressures favored phenotypic and behavioral plasticity, rewarding individuals capable of responding to unpredictability. The result was a lineage primed for expansion into diverse environments, able to exploit niches left by competitors, and poised to radiate rapidly as ecosystems stabilized and diversified across the recovering planet. At dawn, the first stirrings of evolutionary innovation hinted at the dominance these small creatures would claim. Limbs, tails, teeth, and sensory organs were all refined through countless interactions with prey, predators, and the environment. Behavioral strategies, from foraging and nesting to social signaling, were continuously honed, producing a resilient, adaptable lineage. Even in marginal niches, these early dinosaurs demonstrated that innovation, patience, and opportunism could outweigh size or brute strength. The dawn of these creatures was not a sudden eruption but a careful, incremental conquest, laying the foundation for the Mesozoic world that would soon belong to their descendants. In the echoes of oceans, forests, and skies long vanished, we glimpse a planet constantly reinventing itself, where life pressed against every boundary, tested every possibility, and endured against unimaginable odds. The creatures of these eras were architects of change, shaping the world while being shaped by it, leaving traces that ripple through time. From the smallest plankton to the first bold vertebrates venturing onto land, evolution's quiet persistence carved a path toward the age of giants. If this journey inspired wonder, feel free to like, subscribe, and share your thoughts below.